I'm not really proud of the slides that you're about to see. I threw them together to satisfy the need. I wasn't sure exactly what to say, but I thought I'd say something about phenology and behavior because I think sometimes that's not often considered as much as they should be in our models. Let me first start out by saying I've learned that insects and mites and ticks have evolved resistance to many tactics that we attempt to treat them with. Of course, there's the insecticides, acaricides. There's the BT crops. There's crop rotation for at least 2 species that we know about, the northern and western corn rootworms. So there's extended diapause for one of those species, and behavioral change and oviposition behavior with another species. There's been evolution of resistance to pheromone-based mating disruption in insects. So the insects essentially are no longer, the males are no longer overwhelmed and distracted by the pheromone that we distribute in the environment to mask the females that are calling. And there's been field-based resistance to a virus, a pathogen in European codling moth to viral insecticides. So, and then of course in the lab we can even find other forms of evolution of resistance to various things, parasitoids, other pathogens. So I think I've often said that if we think of this in terms of looking at the transient dynamics, not just the equilibrium scenario that will ultimately be the end point, if we look at trying to explain and predict the actual timing of many of these events, I believe that combining population dynamics, population genetics, evolutionary biology, landscape ecology, and try to predict all of this for a particular species in a particular environment. It's a very challenging, rewarding, exciting activity. So I'm glad that you're all here to work on that, and I think it'd be nice if even more people took up the challenge and worked on that. So let me just get to my slides. Hopefully you won't get too bored by it. So I'm going to be focusing on 2 aspects that need to be looked at. Obviously, the starting point, initial conditions of genetic allele frequencies, the survival, selection pressure, the dominance of traits, how they're, the inheritance, the expression. Those are the major big 3 factors that determine generally how fast evolution will occur. But the landscape's important, and then of course how the landscape is affecting phenological factors and behavior of the insect, I believe is very important to understand more precisely what's going on. Behaviors are very important, but they're harder to measure compared to mortality. And so often we don't focus on that first. And then, of course, timing of intergenerational processes is often taken for granted, at least at the first round of going over your problems. So I just want to look at, explain some of the examples that I've taken out of the literature. And so if you don't have this knowledge, you can look at some of these papers on your own time and see how they've dealt with some of these factors. And for instance, feeding and egg laying, the timing of the feeding and egg laying in the mosquitoes, and how this is occurring in relation to the timing of insecticide use is believed to be important for maybe slowing down the evolution of resistance to the insecticides in the mosquito population. And so they looked at how there's actually a long time, in terms of a few days, in terms of the life cycle of the mosquito, where you can focus the insecticide and kill the mosquitoes before they actually are laying their eggs. So one interesting topic. Yves Carrier in Arizona looked at adult emergence in crop planting and talked about how that was very important for dealing with the evolution of resistance to the insecticidal cotton. Some other people looked at diapause, the timing of diapause, and timing of insecticide applications, and how that was important in a couple of different insects. One was Helicoverpa armigera, and another one was the Colorado potato beetle. Again, an example of how phenology is very important. Mitchell and Onstead looked at one of those species of rootworm that has evolved resistance to crop rotation. In this case, it's the extended or prolonged diapause, and how that has influenced the evolution of resistance. So in one obvious sense is if instead of the eggs hatching after one winter, if they hatch after 2 winters, not only do they avoid the effect of the crop rotation, so they've evolved resistance to crop rotation, but obviously they're under fewer generations of selection for whatever else you throw at them, because they're avoiding the 1 year's worth of selection. Again, Carrier and Roth, back when he was back in Canada, studied oblique-banded leaf roller, Crostanura rosaceana, and used a model to look at how diapause would be evolving differently because of insecticide applications. And then also how refuges are involved in all that. And now I want to talk about behaviors. There's so many different behaviors that could obviously be important for evolution of resistance and insect evolution in general. I think we barely scratched the surface in terms of what behaviors might be worth measuring. Timing of a behavior is important, and then also with obviously movement, distance moved is very important. And then are we talking about movement that allows the insect to consider the environment a spatial mosaic, or is the movement restricted enough so that the scale is not so large that if one farm is different than another, it's not going to matter for the population genetics and the evolution of resistance. So the relationship of the environment, the landscape, and the scale of the movement is critical for understanding the importance of these behaviors. Behaviors of adults. It covers everything from mating, local dispersal, selection of food, selection of site for egg laying, migration. All those can be important. Obviously mating. I usually, in the old days, 2 years ago, considered mating to be just a simple thing. I mean, it was just mating. What else was there? I mean, but you can break it down into sub-behaviors. And is there some movement before mating? Is there, which sex moves? Do they both move or just one? And there's, I'm not an expert on mating, so I have a lot to learn. Immigration, of course, was modeled from very beginning in the 1970s and 80s by the mathematical modelers that looked at insect resistance management. Fred Gould in the 1980s was one of the first to model behavioral resistance and an avoidance of toxic plants. So we not only modeled the toxicity to the larvae, but also looked at how the adults might evolve behaviors that determine where the eggs are laid. We've incorporated mating into the western corn rootworm models that we have, and Joe Spencer and some of us got together and published this paper recently with a cute title, Isolated Females and Limited Males. So the females don't move before they mate, at least most of them don't move. And so they may be sitting waiting for the male to arrive, but the males only move a limited distance. I mean, it's not we don't assume random mating, we don't assume unlimited movement of the males. So we have to account for all this in the mathematical model. I did a study of insecticidal cowpea and Maruca vitrata in West Africa. And in that case, we considered 3 regions from the Benin and Togo on the south up to Burkina Faso in the north. And the insect is endemic and resides all year round in the southern countries. But in Burkina Faso, it depends upon the migration every year from the south. And so we looked at the various differences in how you would manage the insect and try to manage evolution to insecticidal cowpea, depending upon what country you're talking about.
Sorry, David, the cowpea, is it a BT? What's the
Right. Well, we just learned at the ESA meeting, at least I did, that they're also considering not only BT, but also a amylase. I think it's some kind of inhibitor of metabolism in the insect. And so various groups are trying to offer this insecticidal cowpea or various transgenic cowpeas for use in Africa for these countries. But they don't want them to be wasted and just fall apart in 2 years. They want something that'll be durable. Oviposition behavior. Oviposition deterrence can be modeled. You could just either hypothesize that someday we'll have some kind of repellent or deterrent in the crop, and so how would that affect behavior, and what would be the consequence of that, and how fast would evolution of resistance to the deterrents occur, so that they would just ignore the repellency and just continue to oviposit in the crop. Similar kind of work was done in the Netherlands with the help of Fred Gould, and this was a more of an abstract model that also looked at how adult preference for location in the landscape for oviposition could be evolving. Some field observations. The BT cotton crop in Australia is an important way to help control the Helicobacter armigera. The people in Australia assess the oviposition by the female moths, and they found no change in the relative ranking of the hosts selected by the females. So they're suggesting that right now there hasn't been much evolution. So people are starting to look at this, both obviously modeling, as well as look at some of these behaviors more carefully in the field, as Joe does with his insect movement studies. Quality of the environment. We shouldn't take the environment, even if we say it's a corn environment, we shouldn't say that all corn environment is the same, because sometimes it's irrigated corn and sometimes it's non-irrigated corn. And we find with some insects, their behaviors are affected by the humidity or the conditions in the irrigated corn, and that's different than what we find in rain-fed corn. And so we took advantage of that knowledge and incorporated that into the mathematical model. And now we could look at larval insects, and, the feeding behavior, moving from plant to plant, or moving from one plant tissue to another plant tissue in the same plant. Someday we might have crops that only express their toxins in certain tissues or organs. So if we want to protect certain things in the plant, maybe we should try to express the toxins only in those tissues that we want to protect. And that would allow more susceptibles to survive on the other kind of plant tissues. And of course, timing of all these is important. we're putting out mosaics. Sometimes, in theory, we say mosaics are bad, particularly if they're mosaics of two different toxins, because it's possible that the insect would just essentially evolve resistance to both simultaneously, and there wouldn't be redundant killing. But you know, with often what we're doing anyway is creating a mosaic, even if we're not trying to. We have commercial seed blends. That's a mosaic of refuge and BT crop. We have seed saving by farmers in certain countries and just cultures that would allow a mosaic to be planted the next year by accident. And then pollen dispersal, contamination, creating a mosaic of expressions in these seed mixtures or non-seed mixtures. And so some of us got together and published a paper called Seeds of Change, Corn Seed Mixtures and Resistance Management, and looking at the variety of issues that have to be addressed when you really focus in on the landscape and what happens when you work with a seed blend. Throwing in some classic papers here in case you're not aware of them. Mallet and Porter in 1992, followed by Tvashnik in 1994, looked at this issue of seed mixtures and refugia in an abstract way, but still a very relevant way. Cross-pollination, couple of papers here. Again, worried about is it possible that seed-feeding insects will be affected by different expression of the toxin in seed that's been cross-pollinated between refuge and BT crops. And so work out of Arizona, looking at BT cotton, and work out of my group in Illinois on European corn borer on corn. And I'm not summarizing the results of these studies because they're complex. Depends upon the specifics of each insect. This case, we found that it wasn't that important because the percentage of larvae that actually go to the seed, the kernel of corn, is low, and for other reasons. But that's one thing. But if you have an insect, the result might change if you have an insect that has all the larvae end up on the corn seed or any seed if there's cross-pollination. So you have to account for as I said, the behaviors and characteristics of each species and each crop before you can draw a conclusion. This is a generalization of some things that need to be taken into consideration when you look at seed mixtures and behaviors. The timing of events, the harm that is done at each location where the insect is feeding, the probability of movement, and the likelihood that there's asymmetry of movement. There might be more or less movement off of a BT crop than off of a refuge or non-BT crop, for instance. And you need to understand that it's not always going to be the same. Larvae can have many events at which they move. They can, with no larval movement, seed mixtures have no risk and several benefits, and they might have several benefits concerning IRM because of the adult behaviors, allowing the greater mixing of the adults. If only one event occurs in which the larvae move, and if the neonates move prior to feeding, then it is possible that susceptible individuals incur no harm that is not also experienced by heterozygotes during and after movement. And then movement as a neonate without feeding is a characteristic of some corn pests. So unless the probability of moving away from a non-BT plant is high, risk is not likely to be high. And as I said, there could be asymmetry. And as I said, the complexity of the issue makes it difficult to generalize. So I showed you those classic papers that were attempting to generalize, but if you look at them carefully, you'll recognize that they did only model certain scenarios and not all possible scenarios. This review does not cover all modeling behavior. People in this group right here in this room have modeled behavior in a variety of other ways that I haven't covered. One more thing, behaviors of humans. If we really are going to talk about management and not just the biology of insect evolution, then we need to account for behaviors of humans, which is a whole another complex issue. So conclusions. We haven't modeled all examples of behavioral resistance. We need to work together so that we can focus on these behaviors. Behaviors can influence IRM, whether they've evolved or not, but we also have to recognize that if there's enough selection pressure, the behaviors themselves will evolve. And timing is important, of course. That's it.